Hello everyone, my name is Moaaz Khokhar and I am affiliated with Cosby Lab at Koch University. In this work, we investigate the potential of incorporating large language models into machine learning and deep learning approaches in order to enhance the prediction of allosteric sites in proteins. Allosteric is the process by which binding at one site perturbs a distant site, rendering it a key focus in the field of drug development with its substantial impact on protein function. Several tools have been developed such as Allopred, Allositro, Spacer, and Parser that leverage normal mode analysis, molecular dynamics, and machine learning approaches. However, according to our knowledge, none of them have leveraged large language models for this problem. This research investigates the potential of incorporating large language model features into machine learning and deep learning approaches based on the fact that the large language models such as ProdTrans based on BERT architecture effectively capture the spatial relationship among residues. Our approach provides promising results and contributes to research on enabling identification of allosteric sites for drug discovery and protein engineering. We collected the dataset from Allosteric database, which comprises of 1944 proteins. We performed some pre-processing steps, such as we removed proteins that had more than 25% sequence similarity. Moreover, we put a threshold of 3 angstrom on the resolution of the protein. These were the same steps that were performed in the previous research by Tau group. They have an automated script for these pre-processing steps. Furthermore, we processed these proteins by the F pocket that gave us 12,486 pockets. As done by the Tau group, we also considered a pocket as allosteric if it has at least one allosteric residue and or it is nearest to the center of mass of the protein. It can be seen that our dataset has a huge class imbalance, and our allosteric sites or the positive samples only account to 6.61% of the whole dataset. In this work, we fine-tune the large language models, which is based on the BERT architecture. This model takes the sequence of the protein and it predicts whether a residue is allosteric or not. It gives a 1024 dimensional feature vector for a residue. Furthermore, F pocket was used to extract pockets from each protein. It gives a 19-dimensional feature vector for each pocket detected. Pockets extracted from F pocket combined with the features extracted from the large language model were used to train different deep learning and machine learning models, including graph neural networks, XGBoost, SVM, and AutoGluon models. Here I would like to mention that AutoGluon is an AutoML library that tries several machine learning models in order to enhance prediction. Each residue in a pocket is mapped to each residue from the LLM, and only the relative pockets residue vectors from the LLM are concatenated with the pocket features. This is the architecture of our work. First, we give a PDB of a protein to the F pocket, which extracts pocket from that protein. Each pocket has a 19-dimensional feature vector. Moreover, each pocket has 3D structure, which is comprised of residues with their XYZ coordinates. Moreover, we give this sequence of this protein to the fine-tuned ProdBERT model, which gives us the feature vector for each residue in this sequence. We create graphs of each pocket such that each node in this graph represents a residue in the pocket, and each node has a 1024 dimensional feature vector that we take from the ProdBERT model. This graph is then fed to the graph neural network, which predicts whether this graph or pocket is allosteric or not. Moreover, we combine these features from the F pocket and the language model, and then we feed these features to the XGBoost, SVM, and AutoML models. And these models are trained on these features in order to predict whether this pocket is allosteric or not. This is the architecture of our graph neural network. It starts with the transformer convolution layer. This is the same transformer that was proposed by attention is all you need. It is based on that transformer. And after that, it is passed to the ReLU batch normalization and pooling layer. And this block is repeated 5 times. After this block, we have three linear layers and sigmoid layer, which gives us the probability whether a pocket is allosteric or not. Our evaluation parameter is F1 score because we have a huge class imbalance. And here P denotes Peng tau. We compare our models to the Peng tau models because they have the recent research in this area. Our models incorporate language model features, and it can be seen that our models in the case of SVM, AutoML, XGBoost, and GNN, they all surpass the previous works. However, in the case of ensemble models, that is comprised of XGBoost and GNN from the tau group, it still has the highest F1 score. But in the case of standalone models, our models surpass their F1 score by incorporating the language features. In the end, I would like to thank my advisors, Atila Gursoy and Özlem Keskin, for guiding me through this research, and I also thank you for listening to my presentation.